Welcome back to the deep dive. Today we are tackling the cell, the absolute basic unit of life itself.
But this isn't going to be just a high school biology review.
No, not at all. We've synthesized a huge stack of sources here, everything from core academic texts to some really advanced papers on polymer physics.
And the goal is to show you the cell not as just some simple bag of goo, but as a hyper-efficient, physically organized factory.
Exactly. Our mission today is to get past the anatomy list and really understand the integrated systems, how things connect, how energy gets made.
And, maybe most surprisingly, how the 3D shape of your entire genome is governed by, well, physics, not just complex biology.
It's a complete systems architecture tour from the ground up.
That's right. The real genius of the cell isn't just the number of parts, it's how precisely they're placed and how they interact.
Okay, let's unpack this.
Where should we start?
Let's start with the two big solutions life came up with. Prokaryotes versus eukaryotes.
Right, the fundamental split. It's really a story of simplicity and then complexity built right on top of it.
Prokaryotes being the simple ones.
The originals, yeah. Archaea and bacteria, the structurally, well, simple. They don't have all those internal compartments.
And eukaryotes, that's us, plants, fungi, we took that basic plan and just ran with it.
We introduced massive internal organization. And the first clue as to why is just the sheer size difference.
This was a real aha moment for me in the sources.
It's huge.
Yeah.
Eukaryotic cells are typically, what, 10 to 50 times larger than prokaryotes?
So we're talking about maybe 1 to 5 micrometers for a bacterium?
Versus maybe 50 micrometers for one of our cells. And when you get that big, you hit a massive physical problem.
Diffusion.
Diffusion time, exactly. You just can't get nutrients or signals across that much space fast enough. It's too slow.
So all the complexity, all the organelles and membranes in our cells, it's basically an engineering solution to being big.
It's the perfect way to put it. Compartmentalization solves that surface area to volume problem. You create all this internal membrane, and suddenly you have more workspace for reactions and much shorter delivery routes.
But even with that huge difference, the sources point out 4 things that every single cell has. No exceptions.
The non-negotiables of life, right? Every cell has to have a plasma membrane.
The barrier between inside and out.
A selectively permeable one, yeah.
Yeah.
Every cell has cytosol, the liquid interior.
Okay.
Every cell contains chromosomes, the DNA blueprints, and crucially, every cell has ribosomes.
And that last one, the ribosome, is so important for understanding prokaryotes.
It is. Why is that?
Well, because it's not membrane-bound, right?
Exactly. The defining feature of a prokaryote is the lack of membrane-bound organelles. No mitochondria, no Golgi, no nucleus.
But they can have ribosomes because a ribosome is just RNA and protein, no membrane.
So they can make proteins. Their DNA, which is usually just a single circular chromosome, it just sort of flutes in an unbound area we call the nucleoid.
Okay, that sets the stage perfectly. So let's jump into the eukaryotic factory, the world of compartments, starting with the control center.
The nucleus.
Yeah.
Usually the biggest, most obvious organelle you can see.
And inside, you've got the DNA, but it's not just a tangled mess. It's organized as chromatin.
Right, which is the DNA wrapped neatly around proteins called histones, and the whole thing is enclosed in the nuclear envelope.
Which, the sources stress, is a double membrane, 2 layers.
And that envelope is supported from the inside by a protein mesh called the nuclear lamina. It gives it shape and strength.
And deep inside the nucleus, there's the nucleolus.
Which is the ribosome factory. It makes the ribosomal RNA, linking the control center right back to protein production.
This theme of connection seems to be everything. It defines what the texts call the endomembrane system, or EMS.
It does. The EMS is this whole network of membranes. The nuclear envelope, the ER, Golgi, lysosomes, the plasma membrane itself.
They don't all have to be touching, but they're all related. They pass materials back and forth using vesicles.
Right. But it's interesting, only 2 parts of that huge system are physically continuous, actually touching.
The nuclear envelope and the rough ER.
Exactly. And that connection tells you something. It highlights this constant, urgent need for communication between making the RNA message in the nucleus and translating that message into protein at the ER.
So the rough ER is, well, it's rough because it's covered in ribosomes.
Studded with them. Its main job is to make proteins that are going to be secreted from the cell or embedded in a membrane somewhere. It's also the cell's main membrane factory.
So if continuity is so important there, why break up the rest of the system? Why use all those little transport vesicles for everything else?
That's a great question. It's because the next steps in the process require very different, very specialized chemical environments.
Ah, so you need separation.
You need separation. If the rough ER is the main assembly line, the smooth ER is the specialized chemistry lab next door. No ribosomes.
So what does it do?
All sorts of things. It synthesizes lipids, oils, steroids. It performs detoxification, which is a huge job in your liver cells.
How does it do that?
It essentially adds hydroxyl groups to toxins, which makes them more water-soluble.
So you can get rid of them.
So you can just flush them out of your system. It also stores calcium ions, which is critical for muscle contraction.
Okay, so once the cargo is made, proteins from the rough, lipids from the smoother, it has to ship out to the Golgi apparatus.
The post office, as it's often called.
But that sounds too passive. It's not just sorting mail, is it?
No, not at all. The Golgi is an active assembly line. It's made of these flattened sacs called cisternae. Vesicles arrive at one side, the cis face,
The receiving dock.
the receiving dock.
Yeah.
And as the cargo moves through the stacks, it gets modified. Sugars are added, maybe turning a protein into a glycoprotein. It even manufactures some of its own macromolecules, like pectins in plant cells.
And the way things move is just, it's so physical, the membrane itself pinches off.
Like a soap bubble, these little spheres called transport vesicles pinch off, move through the cell,
Often walking along those microtubule tracks we'll get to.
and then they fuse with the next target, like the Golgi, delivering the cargo inside.
So what about stuff that needs to be destroyed? The cell's garbage disposal.
That's the lysosome. It buds off the trans face of the Golgi, the shipping side.
And it's basically a bag of dangerous enzymes.
Very dangerous. They're hydrolytic enzymes that work best at a really acidic pH, around 5.0. So the membrane has to keep them contained.
Right, because the rest of the cell is about pH 7.2, so if they leak, they just get inactivated. A great safety feature.
A crucial one. And the lysosome is involved in two key processes. Phagocytosis is one.
Fusing with a food particle to digest it.
The other, which is maybe more interesting, is autophagy.
Self-eating.
Literally,
That's
it's the cell's recycling program. The lysosome will engulf a worn-out mitochondrion, break it down, and salvage all the molecular parts for reuse.
Constantly cleaning house. Okay, before we get to the energy systems, let's clear up the big differences between animal and plant cells.
Right. Three major things plants have that animals don't. First, the rigid cell wall, made of cellulose. It gives them a fixed shape.
Second, chloroplasts for photosynthesis.
And third, that massive central vacuole. It can take up 90% of the cell's volume. It stores water, maintains pressure. It's key.
And on the animal side, what do we have that they generally don't?
We have centrioles, which are involved in cell division, and lysosomes are much more prominent.
But the big misconception, the one that always comes up,
Is about energy. People think plants only have chloroplasts.
which is wrong.
Totally wrong. Plant cells have both. They have chloroplasts, and they have mitochondria.
Because they still need to make ATP when it's dark or in parts that don't photosynthesize, like roots.
Exactly. Mitochondria are basically universal for eukaryotes.
Which brings us perfectly to those two energy converters and their mind-blowing origin story.
The endosymbiont theory. This is just one of the most beautiful ideas in all of biology.
Pioneered by Lynn Margulis and others. The idea that we are hybrid organisms.
We are. The theory says that a very long time ago, an early eukaryotic ancestor engulfed a smaller prokaryotic cell.
But instead of digesting it, it formed a partnership.
A permanent mutualistic partnership. The first event was engulfing an oxygen-using bacterium. That little guy became the mitochondria.
Giving the host cell a massive advantage by unlocking aerobic respiration. So much more ATP.
An incredible energy advantage.
Yeah.
And then later, in the line that led to plants, a second engulfment happened.
This time a photosynthetic bacterium, like a cyanobacterium.
And that became the chloroplast.
And what's so powerful is the evidence for this is written right into the structure of our cells today.
Oh, it's overwhelming. First, look at the membranes. Both mitochondria and chloroplasts have a double membrane.
The inner one being the original bacterial membrane and the outer one from the host cell that swallowed it.
That's the idea. Second, look at their ribosomes. They make their own proteins, and they have their own ribosomes to do it.
And they're the smaller 70s bacterial kind, not the 80s kind found in the rest of the cell.
A huge tell. And the smoking gun really is the DNA.
They have their own DNA.
A single circular chromosome of DNA, just like a bacterium. And they reproduce on their own schedule.
By binary fission, like a bacterium.
By something very similar, yes. You can literally watch them dividing inside our own cells. We are powered by ancient captive bacteria.
It's incredible. Okay, so we've built the factory, we've powered it. Let's talk about the internal infrastructure, the cytoskeleton.
Right. This is the cell's support system, but it's incredibly dynamic. It's not a fixed skeleton like ours.
It's constantly being built and torn down.
Exactly. It provides shape, it anchors organelles, and it's the highway system for all internal transport.
And it comes in three main types of fibers. Let's start with the biggest, the monorails.
Those would be the microtubules, hollow rods about 25 nanometers thick, made of tubulin.
And their big job is transport.
That's their main dynamic role, yes. Motor proteins, like dynein and kinesin, literally walk along these microtubules, carrying vesicles and other organelles as cargo.
They also form the core of cilia and flagella, right?
They do, in that classic 9+2 arrangement. And they're essential for separating chromosomes during cell division.
Okay, moving down in size, we have the intermediate ones.
The intermediate filaments, about 8 to 12 nanometers. And these are the stable ones, the rebar of the cell.
So they're not for movement.
No, their job is to bear tension and just hold things in place. The nuclear lamina that we mentioned earlier, that's made of intermediate filaments that give the nucleus its shape.
And finally, the thinnest, most dynamic fibers.
The microfilaments, or actin filaments, only 7 nanometers thick. A double-twisted chain of actin protein.
And these are the real movers and shakers.
They are. They are essential for muscle contraction, working with myosin. They cause cytoplasmic streaming in plant cells, and they allow cells to crawl, amoeboid movement, by rapidly building and extending the network.
Okay, so we've built the entire factory. Let's zoom back into the nucleus for the final concept, which is, it's a bit of a leap, the physics of genome organization.
It is, yeah. This is from some very specialized source material in polymer physics. We usually think that DNA organization, where chromosomes are, how they loop, is all driven by complex biological signals.
But this model suggests something different.
It suggests that this organization can just spontaneously arise as stable thermodynamic states.
So you're saying the structure isn't always being actively pushed into place, but that it just falls into the most stable, lowest energy arrangement.
That's the core insight. The cell can control this organization by just tweaking a few key factors until they cross a threshold. It's like flipping a thermodynamic switch.
A switch with three main variables.
Right. First is affinity, the binding energy between some linking molecule and the DNA. If it's too weak, loops fall apart. If it's strong enough, they become stable.
Okay, affinity. What's the second?
Concentration, the concentration of those linking molecules. If you have enough of them above a critical level, the chance of them forming multiple bridges skyrockets and just forces the DNA to collapse into a compact, looped territory.
And the third is just having enough places for them to bind on the DNA itself.
The binding site fraction, yes. You need enough anchor points.
The sources use this term, kDollars, to talk about binding energy. They say these structures can be stabilized by energies as weak as like 2 kDollars. What does that actually mean?
That's a great point. So kDollars is just the unit of thermal energy. It's the background jiggle and noise of molecules bouncing around because of temperature. A bond of whole kDollars would be broken instantly by this noise.
So 2 kDollars is barely stronger than the background noise.
Barely. And that's the amazing implication. The cell doesn't need super strong chemical locks to organize its genome.
It uses quantity over quality.
Precisely. It achieves stability by having a high concentration of pretty weak linkers, which means the cell can control the entire physical layout of its DNA just by turning protein expression up or down.
Just by controlling concentration.
It's an incredibly robust and simple switch, and the physics even explains how different chromosomes arrange themselves in the nucleus, sometimes forming specific geometric patterns just based on the competition for these linking molecules.
Wow. Okay, so to recap, we've gone from the simplest prokaryotic cell
to the intricate, interconnected factory of the eukaryotic endomembrane system.
We've uncovered the hybrid bacterial origin of our power plants,
Mapped out the dynamic highways and support beams of the cytoskeleton.
and landed on this idea from physics that the 3D structure of our very genome is governed by these simple, spontaneous, concentration-driven switches.
An incredible journey from a single membrane to the physics of life itself.
It really is. And it leaves me with a final provocative thought for you to chew on. If the complex organization of life's machinery is governed by these simple laws of polymer physics, these thermodynamic switches of affinity and concentration, then how much of what we call cellular decision-making when a gene is exposed, when chromatin condenses, is really an active biological choice, and how much is it just molecules, spontaneously settling into the most stable physical state that the local environment allows?